We've talked about absorption and distribution, and now we're going to talk about how the drug exerts its effect in the body. This diagram shows the ADME part. Now, we haven't talked about the metabolism or the excretion part, but pharmacokinetics, that is part of that, how a biological system, how the drugs move through the body. But pharmacodynamics is how the activity that the drug exerts on the body. So this diagram reflects that. So in order for a drug to exert an effect on a tissue, the tissue has to have a specific protein on the surface of the cell or within the cell in order for that drug to react with it. This is called a receptor, and a receptor and a drug are kind of like a lock and a key. The drug sits on the receptor in such a way that it can stay there for a while. It may stay there for a long time or have a strong affinity, is what we call it, for the drug to bind to the receptor, or it may simply, touch on it. Something may happen in the cell and then it may leave. Eventually, the drug molecule leaves the receptor, but whether it exerts an effect or not, that will indicate what kind of drug that it is. So, the receptor and the drug have their shape that corresponds with each other, and that allows it to bond with it. And it will create a, a reaction in the cell or a cell change. It may release a protein. It may allow ion channels in the cell to open up and ions to come into the cell and affect some kind of change. It may start a cascade reaction. All cells probably have receptors, but they don't all have the same receptors. Some of them have different receptors. And different receptors as in many receptors. So with that, different drugs can affect different cells in different ways. Even if we get the concentration of a drug in near a tissue that does not have receptors on it, that tissue is not going to react to the drug. The drug has to have the receptor that is specific for that particular type of drug. So here is a little diagram just showing multiple receptors. So here's drug A, and it's sitting on one of the drug A molecules is sitting on a drug A receptor and exerting an effect. There is another receptor for drug B, and it exerts its own effect. So we can have two different effects. They may counteract each other, they may enhance each other. One may do nothing and the other may have an effect, but those are receptors, and that's what happens with the drug molecules. Now, if the drug exerts, after it sits on the receptor, it exerts some kind of activity in the cell, like I said, releasing a protein, opening ion channels, starting another cascade of events, that is an agonist. It has the agonist is a drug that together with that receptor exerts an intrinsic activity. Intrinsic just means inside of the cell. An antagonist is a drug that does not exert an activity in the cell. It may just sit on the receptor and occupy that receptor. So here is a diagram of that, an agonist drug sitting on this receptor. We have an effect on the cell. And over here we have an antagonist drug. It's sitting on the same receptor. It's just not exerting an effect, but it is exerting, by not exerting an effect, it is basically blocking this drug from sitting on this receptor. So it may decrease the effect on the cell if there are a number of these receptors that are blocked and the agonist drug cannot get to the receptor. So antagonists can have uses, and we use them typically what they are used in veterinary medicine is as reversal agents. So they may, come in and block the receptors. Let's say we give an anesthetic or a sedative type injectable sort of drug, and um, that drug exerts an effect on its receptors. But then there's a point where we want that animal to wake up, and we can use a reversal agent, and that is an antagonist. It basically goes in and it will now go to those same receptors and effectively block them because it doesn't exert an effect, but it does sit on the receptor. Bear in mind that the agonists and antagonists are constantly, attaching and then detaching to the receptors, and the one that it has the most concentration at the time is the one that's going to exert an effect, whether it's the intrinsic activity or whether it's, in the case of an antagonist, in blocking that activity. So a very common antagonist that you probably have all heard, is in the news these days with our opioid crisis that's going on, is a drug called Narcan. It's also called naloxone. That particular drug is an antidote to opioid overdose. And it basically goes and blocks those opioid receptors, not allowing that drug, the opioid drug, to take effect and starting to reverse it. So, a competitive antagonism. This is called it's called a reversible, surmountable type of antagonism. So those might give you some other clues to what it is. It's basically the idea that if you have two receptors and they can, or two drugs, I'm sorry, two drugs that act on a particular receptor, and you put in more of drug A, then drug A is going to have the predominant effect. If you put in drug B, it's going to have the predominant effect. So that is why if we are giving a reversal agent to a drug that we gave a little while ago, and let's say that is an agonist that we've given a couple of hours ago, but now we want to reverse the effects and we give an antagonist, that is going to be very effective in reversing as long as they're pretty equally competitive to the same receptors, because there's probably not as much of the agonist around, and the antagonist is the one that's going to have the effect. It's reversible because, or surmountable, because it's simply, if we added in the agonist again, that is going to take predominance and that will have the greater effect. So non-competitive antagonism is a slightly different take on this. So what it is when we have an antagonist that has a great affinity for the receptor and occupies it. It may change the shape of the receptor. It may change the availability of the agonist drug to sit on that receptor. And it appears as though it is a permanent type of reversal agent. So it's called irreversible or insurmountable antagonism. It just has to do with that drug having a great affinity for the receptor. And this is an illustration of that, where we have an antagonist drug that would normally sit on this receptor, but because the antagonist drug has basically changed the accessibility by not allowing that agonist to fit into the shape it needs to, the sort of lock, then it's effectively blocking that agonist drug. This is an illustration of where the antagonist drug has changed the receptor shape itself. It's changed it so that it can fit there, but you can see the agonist drug has a different conformation, so it can't occupy that receptor. So it effectively blocks, it makes it look as though it can't be reversed, and in time, remember, these antagonists don't sit there forever, but they may be there for long enough that it effectively does not allow the other drug to act at all and blocks it. So that is non-competitive antagonism. So, looking at, I'll kind of ask these questions. Drug concentration, does it affect competitive antagonism? Well, it does because we can allow either one, we can allow them to compete with each other. If we give both of them, we typically have to give one in more concentration for it to kind of take the lead in what is going on. So we can simply reverse an effect with another drug by adding more concentration of that drug. With non-competitive antagonism, that concentration does not affect how that antagonism works. It simply has a greater affinity, or it changes the receptor so that the other drug cannot have effect. And it doesn't relate so much to concentration. So there is something called a partial agonist antagonist type of drug, just to make it even more confusing. And butorphanol is one of those drugs. Your book gives an example, and it's basically talking about this drug. It just doesn't name it. And it is both an antagonist and an agonist. So it is used, it is a lighter analgesic, and also induces some narcosis, so it makes the animal a little drowsy. Let's say we've given an animal an opioid drug, such as morphine, to give the animals come out of surgery. We want to make sure it's not feeling pain, but it makes the animal too drowsy. It starts to affect its respiration, which opioids can do. They depress the respiratory system. So let's say we want to reverse that, but we still need some pain management on board. And butorphanol is one of those drugs that can basically block or antagonize the morphine, and but also sitting on that receptor, those same receptors, provide some analgesia. So that's a partial agonist antagonist. So it somewhat reverses the effect of something that we want it to reverse, but it also has its own effect. So lastly, there are ways in which drug exert their influence in the body not needing receptors. So one of these is mannitol. Mannitol is basically a large sugar composed of large sugar molecules. And when we give this drug, we typically give it IV, and it draws water out of different places. If we have a lot of swelling in some place, it will draw the water out of it. It is freely filtered in the kidneys, and so it ends up in the nephrons, and it keeps the water with it, and so the water leaves the body in the form of urine. It's simply a sugar. So the way it acts is it is an osmotic type of diuretic, if you will, water reducer. So it is by its concentration and staying in the blood, and then being freely filtered in the kidney and leaving the body that way. It brings water with it. It's just a matter of concentration. It's higher in the bloodstream, it draws water out of the tissues, and then it's eliminated from the body. A second type of non-receptor action is a chelator. We use chelators in these purple top tubes when we collect blood if we don't want it to coagulate. So we have a chemical called EDTA that basically combines with the calcium and does not allow the blood to clot. It's a simple chemical reaction. It's just a binding reaction. We can use some drugs to eliminate things like lead in the body. If we've had an exposure to lead, we introduce a chelator that binds with that lead and excretes it from the body. And then lastly, antacids. We'll talk about these when we get to the GI tract. And antacids are simply, as they kind of imply, and they'll neutralize acid. We typically use them in the stomach to neutralize stomach acid when that is having a bad effect on the stomach. And it's a simple chemical process of neutralizing the acid.